Hello, everyone. Good day, and welcome to another episode of ALS Clinical Trials Unboxed, where we give a little window into clinical developments in ALS. My name is David Taylor. I'm the Chief Scientific Officer at ALS Canada, and it's my pleasure to host this episode. I want to say a big thank you to everyone who submitted questions in advance. If you did submit a question about the specific topic, we will make sure that if it doesn't get asked in this short half hour, that we will get back to you with an answer on those. If you provide a more general question about clinical trials, and I know some of you did, those are important questions to us. They really do matter, and we will be happy to answer those questions. We actually have a research and clinical trials 101 Q&A drop-in that happens monthly. We urge you if you can come to that. The next one will be Wednesday, May 21st at 1 p.m. You can come talk to our research team. Be happy to answer those questions, or you can always email us at research@als.ca. Your questions are important to us, and we're certainly here for you. Without further ado, it is a huge pleasure to be able to introduce the Senior Clinical Development Medical Director at Novartis, Dr. Ron Miller. Dr. Miller actually grew up in Ottawa and did degrees at McGill. So for the Canadians in the audience, that's really awesome because he has moved on to an incredible career in clinical development and other areas, epidemiology, etc. So we're really great to have, or glad to have him here. He's going to talk about the ASTRALS clinical trial, phase 2 clinical trial, which is going to talk about VHB 937, a really intriguing biological pathway that this is focusing on. So hopefully we'll be able to touch on that a little bit afterwards in the Q&A. So please do submit your questions live, either in the chat or in the Q&A, and we will try to have a look at those. But without further ado, I pass over to Dr. Miller. Thank you so much for joining us, and the floor is yours.
Thank you very much, David. I hope everyone can hear me. And will you be sharing the slides, or will I need to do that?
We can hear you, and if you're able to share the slides, that would be great. But otherwise, I can look to my colleague in case she has them.
I had to restart my computer, so I
No
don't
problem.
have them quite
No
pulled
problem.
up, but
We can wait on that. That is not a problem.
okay. Sorry. Sorry.
No problem at all.
Here, I think I have them here.
Wonderful.
Um,
And I'll stay on to let you make sure we know that we can see your screen, and then I'll jump off and come back.
all right. Sorry. Just double check. Sorry, I'm having some.
It's okay. And now you're saying sorry a lot, so you're proving that you did grow up in Ottawa. So we're
I did grow up in Ottawa. For everyone who's listening, that part is true.
well, you come in by the
Okay, so
stories naturally. It's all good.
all right. So I will share my screen. There it is.
Okay.
And hopefully you can see that.
It's starting up. We can see it. Yep. And it's not in presentation mode, but if
It's not?
no, we can see the tops and the bottom of your screen. So if that, but I mean, we can see your slides if that's helpful. I mean, it will work just fine the way you've got it if you advance your slides as you've got your cursor.
Okay, I'm going to keep it this
Sure.
way if that's okay.
Not a problem. All over to
Um,
you now. Thank you so much for being here.
Okay, so thank you very much. Sorry for the technical delays. I think that's become par for the course, unfortunately. So it's my great pleasure to be able to talk about our ongoing clinical trial in ALS that is called the ASTRALS trial. As just an additional background, I'm the medical monitor on this study, which means essentially that I'm responsible for the medical safety, but I work on a big team of Novartis colleagues, and It just so happens that because I grew up in Ottawa, I volunteered to do this presentation. I should say I insisted that I would be the one to do this presentation, but there is a huge team that is working at Novartis to implement this trial successfully. Um, sorry, my slides are. I'm going to have to pause for a second because I'm having some difficulty, as you can see with
No
my
problem.
screen. Um, I apologize. Do you have the slides? Did I send them to you? Because if not, I hope I'm no longer sharing because I'm going to have to
You're
find
not
them in a
sharing
different
anymore.
format.
No problem. And I'll look to my colleague in case we do have them. No, we don't have the slides. So not a problem. We are happy to wait on this. And while you're doing
I'll
that,
just,
oh, yep.
yeah, I apologize. I'll try to do this as quickly as I can.
It's no problem. I'm happy to tap dance here and actually talk about why I think this is a really intriguing clinical trial. So if it helps to set it up a little bit, I mean, for years in ALS, we've really focused on taking the cells that surround the motor neurons and trying to dampen a negative sort of what we thought was sort of a negative inflammatory effect that is clearly happening. And that has been negative in most of all the clinical trials that we've tested to date. And so what's really I find intriguing about this one is that the idea is to try to take those cells and actually bring back some of their normal potentially protective function that they have, throughout the duration of our lifetime. So in this case, what you're going to hear about, I'm sure, is TREM2, and as you're trying to give this back to the cells that support motor neurons, microglia, then the hope is that they're going to be able to reestablish a function that we've been missing in ALS. And so I find that a very unique pathway compared to what we've tried in previous clinical development, focusing on these types of cells and these types of pathways. So I'm really excited to hear about that part. Um,
Okay, I'm hoping this will be better now.
okay.
Um, um, again, I apologize,
No problem.
and I will share my screen again. Thank you for your patience. So now you should see it in complete.
Perfect.
I just had to download it because for whatever
Okay.
reason, loading it off my internet drive wasn't working.
All good. We can
So
see it.
I won't go into as much of an introduction, and I will go to the next slide. So just in terms of a disclaimer, this is a Novartis presentation, and I'd ask that, I mean, this is really for non-promotional scientific purposes, and everything I'm presenting here is accurate as far as we know it now. And please do not share this beyond, and um, I'll leave the other disclaimers here. So these are our legal disclaimers. But okay, so um, what is the rationale for this? So
Dr.
David
Miller,
gave
we
a bit
actually
of,
lost your slides.
we lost my
You're not sharing your screen
slides.
anymore.
I apologize.
No problem.
Let's try
There we go.
again.
Looks like it's coming back now.
All right.
It's still a black screen for me. If my colleagues see anything different, they'll let me know. They'll pipe in.
It looks good.
Oh, you can see
You
it.
can see
Okay,
it?
it's just me who can't. Okay.
I'm sorry. I hope the
No,
others can
that's
see
okay.
it.
Perfect.
So, all right, let's try. Hopefully the technical difficulties are behind us. So, what's the rationale for this study? So, as David was alluding to when I was struggling with my technical issues, um, Inflammation is believed to be a major contributor to neurodegenerative diseases. And microglia are important cells in the brain that, in response to degeneration, um, they are brought into play to clear dead motor neurons or damaging proteins. And but unfortunately, as neurodegenerative disease progresses, pro-inflammatory signals can induce these microglia to assume a more neurotoxic rather than neuroprotective effect. And this can lead to a detrimental negative cycle in that the cells act more neurotoxic and they cause more damage, which in turn stimulates them. And this is believed to contribute to the disease progression in neurodegenerative diseases. But part of the activation of microglia is regulated by a receptor called TREM2. And activation of TREM2 induces microglia to shift to this neuroprotective phenotype, this to really enhance the neuroprotective effects. We have a antibody called VHB937 that binds to this receptor and stabilizes it while activating it. So essentially it binds, activates, and stabilizes to sort of enhance the effect of this, what we believe is a beneficial pathway for microglia inducing neuroprotection. And we've designed a study to evaluate this in ALS, and this study is already up and running. We have, this is the first time we're testing this antibody in ALS patients, but we do have data from a study in healthy volunteers that showed that it reduced some of the markers of this neuroinflammation that we're trying to favorably alter in our ALS study. So we have some data in humans that suggest it's working in the way we want it to work. So what is this ASTRAL study in ALS? So it's a study to compare the efficacy of this antibody compared to placebo on both function and survival. Function measured by the ALS FRSR and survival measured as overall survival or the need for a ventilator. We have various other endpoints. We're measuring breathing capacity measured by slow vital capacity. We're measuring biomarkers like neurofilaments and various other outcome measures, including clinical scales and other biomarkers. The key inclusion criteria of the study are listed here. The study is only in adults. You have to have an established diagnosis of ALS with symptom onset within 2 years. And there are also some limits on the slow vital capacity and the ALS FRSR score. We have some exclusion criteria. Most of them are if you can't be on another experimental study, and we have some exclusions for significant other diseases, uh, particularly to ensure that because we don't have a lot of experience with VHB 937, we want to make sure the people are otherwise healthy and won't have a lot of potential side effects from the drug. This is a schematic of the study, and it's very, essentially at a very high level. The screening period runs up to 4 weeks, and at the end of screening, if people are eligible, they're randomized in a 2-to-1 manner to receive VHB 937 or a matching placebo. And this randomization period lasts for 40 weeks, over which time you receive the infusions with VHB, which is given as an IV infusion once a month. There are questionnaires that you'll asked to fill out either when you come in once a month to the clinic to get the IV infusion, or there's some questionnaires that would be filled out at home. There are 2 lumbar punctures in the study, one before the first dose and one after the 4th dose. Following this 40 weeks of double blind, everybody then switches over to the active treatment with VHB, and this continues um, for as long as we have people into the study, um, and as long as people, essentially as people choose to continue in the study. Um, at the end, if someone chooses to stop treatment at some point during the open label extension, we have a safety follow-up visit 2 months after just to make sure everything is okay with the participants. We plan to enroll 225 participants at 92 sites worldwide, with 5 sites planned in Canada. And the sites are listed here. And I believe at the present time, the first 4 sites, the 2 Montreal sites, the one in Toronto and the one in Hamilton are active, and the one in Calgary is set to start very soon. So with that, I think that's the last slide that I'd planned to present, and I'm happy to answer any additional questions.
Great. Thank you very much, Dr. Miller. It's that's like a really nice phase 2 trial. I think it's like the return to a true phase 2, because in ALS we've had so many underpowered things that have sort of been considered phase 2 threes. Is this, with more than 9 months of randomized trial and a long-term OLE, is there like an intent to see what, like I'm the first person who doesn't want to talk about efficacy on things that are underpowered, but this is a nicely powered phase 2. Are there thoughts about what hope to get out of this in terms of um, the longer term,
Well,
both randomization and OLE?
I think you point out it is probably larger than some of the other phase 2 trials, and it's difficult to speculate, but the study is adequately powered to show a statistically significant effect if we're I would say in the range that we're really doing something.
Sure.
So if the effect is meaningful, we should be adequately powered to show it.
That's great. Well, and obviously, I think with Novartis being a big company that's done a lot of these over the years, it will be a a good solid assessment as to what the next steps are afterwards. So, we do have a couple of questions in the chat. So I've got one before that. One of the things I ask almost everyone is around target engagement. I know that soluble TREM2 has been found in the CSF increased. Is there uh, a chance that you're looking at sort of seeing if that can be decreased? Is there anything around, recently we've got some new sensitive measures for cryptic exons, and there's been some preclinical work around connecting to TDP43 biology. Is there any of these kind of, but first the target engagement and then some of the more novel biomarkers that are involved, any of those?
So because of the mechanism of action of VHB 937, it actually binds at a part of the receptor that it stabilizes TREM2. So it actually also stabilizes soluble TREM2. So what we see and we've seen in our preclinical models, and we saw in the healthy volunteer studies that I mentioned, is that soluble TREM2, both in circulation and in the CSF, increases.
Okay.
And that is actually the effect that we anticipated.
Okay. So, oh, you
It,
actually see an increase in the
we,
biofluids of the soluble because you can detect it versus when it's shedded off or
so even the shedded component, we reduce the shedding on the receptor. So just in terms of background, what happens in, maybe I'll just for other people who don't understand it perhaps as well as you do, what happens is the receptor is on the the microglia, and what happens is in life it's shed, and once it's shed, its activity, that stops its activity. The, the nature of VHB937 was designed to not only bind and activate, but reduce the shedding. So we reduce the shedding of the receptor that's on the microglia, but the molecule also binds to the to to TREM2, to soluble TREM2 that's already been shed. And in
okay.
so doing, it increases its half-life, so it's not cleared.
I
So what
see.
happens is we see an increase in soluble TREM2, but that's not an, in some cases that could be interpreted as unwanted, but in our case, it's exactly what we expected.
I see. Okay. That's a really interesting way of looking at it in terms of like um helping to assess the target engagement. Okay. Um, so I have a couple other questions, but we also have a couple questions that have been added by recipients. One question we had in advance was whether or not there's any published access to the phase 1 healthy control data or healthy volunteer data. I know there was a presentation at some point, but
Yeah, I think there's no published manuscript yet, but the data were recently presented at several big meetings. And so you could, if you had the abstract book, which is
sure.
essentially published, you could probably find the abstract books online. Um, you could find just the abstracts, but the full details, we anticipate they'll be published shortly.
Great. That's fantastic. And there was a question around whether or not astrocytes are affected by TREM2 agonists. So I think there's some preclinical literature on that. Maybe you could
Yeah, I
talk about
think
that.
there is some. I think they are affected, but certainly they would be more directly affected due to the crosstalk between the other cells in the brain, like the microglia. I
Yeah.
think we would expect some beneficial effect on astrocytes, and this is I believe that the role of TREM2 and how it affects astrocytes is something that's looked into a lot in the Alzheimer's disease literature,
Okay.
because some of these same processes take place.
Yeah, and I think that it's I think there's even literature outside of TREM2 that have shown that if you can restore microglial function in certain ways, that it also can have some effect on astrocytes. And for the audience out there, the astrocytes are the other primary supportive cell of the motor neurons and other neurons in our central nervous system. There are a few of them, but astrocytes and microglia tend to be the two that we focus the most on. So I think the question and the answer by Dr. Miller is really relating to if you sort of help one of them to restore some of its protective function, can you also help the area around it by supporting those other cells to be less detrimental and perhaps more protective? Um, so there was another question in the chat, which I also had as well, which is 92 countries. That's a impressive uh, set of sites. Sorry, 92 sites. My apologies, not 92 countries. That would be really unprecedented. Are you able to allude to approximately like how many countries that is, or is that a wide array of continents, or is it mostly Europe and the United States and Canada area?
Yes, I would say regionally, off the top of my head, I forget how many countries. I should have that statistic, but regionally, it's the United States, Canada, um, Europe, um, I would not just Western Europe, but some Eastern European countries as well, and Asia Pacific countries. So
Amazing.
Korea, Japan, China, Australia.
Amazing. Well, and I know that the International Alliance will be glad to hear that too, because I think that that's, finding ways for, and especially a company like Novartis being able to have a presence in some of those countries and being able to help ALS clinical development in places that are less represented in most trials. That's fantastic. That's really, really great news. Um, so we have I have another question, which is around the 4 weeks. So actually 4 weeks is really like once a month dosing. So then the TREM2 is fairly stable when you phosphorylate, I guess the stabilizing factor that leads to its stabilization, it stays for quite a while.
Yeah, I think based on the data that we have from both, preclinical pharmacokinetic modeling and what we saw in the healthy volunteer studies, um, we believe that the once a month is more than adequate. We have good coverage over that time period.
That's fantastic. There was a question about whether or not anyone's ever overactivated TREM2. Is there a possibility that that can happen, or is it something that, is felt that the stabilization and activation kind of reaches a natural point on its own?
Yeah, it's interesting. There are some individuals who have deficiencies in TREM2, and they've been identified, but we're not aware of what happens with overactivation. There are no models for that. Uh, there, there are no known mutations that cause that. Um, and this study, the ASTRAL study, will really be the first long-term study in humans. So we'll be able to monitor the safety and see if there's any emerging signal.
Sure. And so, as I kind of alluded to at the start, I think one of the things that I find really fascinating about this one is that most of what we've tried in clinical development, and I know that I know that s- Novartis had previously been working on like a CSF1 receptor inhibitor, and that those are really focused a lot on the dampening of the inflammatory response, which we've done since the very first papers that came out that said, microglia bad in ALS, and we got to go and just tamp those down. And then this is coming from that other angle of, well, maybe what we're also have missed all these years is this idea that they play a really important role with their normal function. And if you can restore that. So I was just wondering if maybe you could expound on that from your thoughts, because yeah,
Sure, and I think that's the beauty of this pathway, the TREM2 pathway, in that it doesn't it doesn't decrease the number of microglia. All it does is it changes their, we call it phenotype, but you can think about it mood or temperament. It changes them from the angry, pro-inflammatory um, phenotype to a neuroprotective phenotype, in that
right,
we're trying to push them in the direction of neuroprotection and pull them back from the direction of neuroinflammation. And that's what this pathway does normally. We're just stimulating the pathway in order
right,
to enhance those features.
right. Yeah, and I think that's just what we've been missing because I think we had half of that, but we didn't have the switch back to the idea of with most of what we were trying, I think in clinical development, it's been just trying to dampen the negative effects without thinking about the switch back to the previous. So we're actually very close to time, but I do have one other quick question. As you alluded to, you have a strong team of people working on ALS at Novartis. I know obviously Dr. Bruin is also someone else who's very well known in the ALS field. Are there other things in clinical development at Novartis that are, maybe not ready for prime time, but still kind of thinking about, the ALS space?
Yeah, I'm not sure what I'm allowed to say, but I
Of
think
course.
it's public knowledge that ALS is considered a priority area for Novartis. So I hope that we'll have other things that we can bring to the clinic
That's
before
music to
too
my
long.
ears. That's fantastic. I mean, having a company like Novartis who is, sticking around in the field is really a welcomed thing to hear and appreciated. And really, we're actually at time. So, thank you very much for giving us a window into the VHB 937
Sure.
program. And oh, yep.
I just have
Did
a
you?
question.
Of course.
I see there's some questions that popped up in the Q&A.
Yeah.
Should I just answer them in the Q&A?
Um, so I mean, we can figure out a way afterwards, I think, to answer those for you. I think most of them we covered. A couple
Okay,
of them we can definitely either reach out to you to get, your answer on those and we'll get back to those individuals. Um,
okay,
but I think just for the sake of those people that booked it only for half an hour,
sure,
we'll end it off, and
absolutely.
but we'll make sure that everybody gets the answers that they're looking for, and really appreciate your time in coming today and giving us this overview of it. And for everyone out there, the best way we can make this the best program possible is with your feedback. So when you close down this Zoom, you will get a short survey. If you could please fill that out, it would be super helpful to us with ALS Clinical Trials Unboxed. So thanks again, Dr. Miller, and thank you to everyone who watched. And we'll see you at the next episode of ALS Clinical Trials Unboxed.
Thank you for your time, everybody. Thank you for listening.
Take care.